Welcome back to a Biology A-Level video. In this video, we will be going over prokaryotic cells and viruses. It is important that you know the structure of prokaryotic cells and are able to compare them to the structure of eukaryotic cells. One main difference to know between prokaryotic and eukaryotic cells is that prokaryotic cells do not contain membrane-bound organelles. An example of a prokaryotic cell is bacteria. Prokaryotic cells do not contain a nucleus. Instead, they contain circular DNA, which are not associated with histone proteins. They also contain a plasmid, which is an additional piece of DNA. This is different from the circular DNA. Plasmids, for example, contain genes for antibiotic resistance, which can be exchanged with other prokaryotes. Compared to eukaryotic cells, they contain smaller 70S ribosomes. Remember, ribosomes are not membrane-bound organelles. Prokaryotic cells have a plasma membrane but have mesosomes, which are inner membrane foldings containing enzymes needed for respiration. The capsule is a protective slimy layer which helps to retain moisture and adhere to surfaces. The pili are hair-like protrusions to help attach to neighbouring cells. The flagellum is a tail-like structure that helps the cell to move. Bear in mind, the flagellum, capsule, plasmid, and pili are not always present. The cell wall in prokaryotic cells are made up of peptidoglycans. There are two types of bacteria, gram-positive, which likes to float around outside, and gram-negative, which likes dark places inside. Gram-positive bacteria contain a thick layer of peptidoglycan on their cell wall. In gram-negative bacteria, they contain a thin layer of peptidoglycan with lipopolysaccharides on their surface too. So, in order to tell which bacteria you are looking at under a microscope, you need to stain the bacteria. As a side note, this is used in the real world to help guide which antibiotics to give someone when they have a bacterial infection. In the staining process, first a violet stain is applied and then washed, and an iodine solution is applied and washed again before alcohol is added. The final step is using a counter red stain. Gram-positive bacteria will stay violet due to the thick peptidoglycan cell wall, whereas gram-negative bacteria will stain red. Now, let's talk about viruses. These are non-living organisms, hence in order for viruses to replicate, they need a host. Viruses contain a nucleic acid genome containing either DNA or RNA. This is surrounded by a protective protein coat called a capsid. The virus has an outer envelope which is a lipid coating. As I was saying, the viruses need a host to replicate, so the first thing it needs to do is attach to a host cell and enter into the host cell. The virus will then inject its viral genome into the cytoplasm and use the host machinery to produce its own viral proteins. In order to release the viruses made, lysis of the host occurs. Hope you found this video useful, and please give this video a like. If you want to see more videos like this, please subscribe.